Section 6, the cell cycle. So this is a sketch of the cell cycle, and this has to be memorized. So here's the story. This is pretty important. Here's a brand new cell just made from another pre-existing cell. But anyway, here's this cell, and the first thing it does is kind of hang out. The cell matures. So this is called the G1 phase, also called gap 1. And the cell is just doing the things cells do. The cell's growing and making more organelles and just doing all the things that the cells do. This next phase is the S phase, or the synthesis phase. And this is the phase where the DNA is copied. This whole entire thing, everything, whoops, all of this in blue is called interphase. And interphase, inter meaning it's between mitosis. So mitosis is cell reproduction. And of course, it's asexual, right? So maybe cell division might be a better name. Cell division, cell reproduction, but make sure you remember it's asexual. So in other words, your skin is just making more skin. So cell division is probably a better term because this implies you're making a new organism. Anyway, this whole blue part is not that. You're not dividing a cell. You're just one cell and you're just growing and copying your DNA. G2 or gap2 is just when the cell is hanging out again and this time getting ready for cell division. Now we're in mitosis, or mitotic phase, means the same thing as mitosis. And so mitosis is this part, and then cytokinesis means you're splitting the cell itself, whereas mitosis means you're splitting the DNA. Your DNA is pretty complicated and it has to separate out precisely. It's not like a bacterial cell where you just have one, you copy it, you separate it. You've got 46 chromosomes. They've got to be separated perfectly, or else one of the daughter cells will get some of the DNA and the other daughter cell maybe will get more of it or whatever. You've got to get it perfectly divided. And so there are, I don't know if you don't really have to memorize this. There's prophase, metaphase, anaphase, telophase. So 4 phases, and I usually just remember PMAT, and that helps me to get them in order: prophase, metaphase, anaphase, telophase. And then, so this is all the nuclear division, how you separate out the DNA. Nuclear or DNA division. Cytokinesis is when you're actually separating out the cell, the goop, the cytoplasm. So cytoplasmic division. That doesn't have to be perfect. If you get a little more on one and a little less in the other for cytokinesis, well, if you've got, 100 mitochondria here and 115 mitochondria here, no big deal. More mitochondria can be made as long as you have some to start with. So what we're going to do now is spend some time on each one of these, and then we're going to spend some time on this. So phases of the cell cycle. The first phase is called interphase, and there are 3 sections of that. That's this whole thing in blue. So G1 is the gap 1 phase, and that's the primary growth phase. The cell's maturing, making more organelles, all that stuff. That's here. The next phase is the S phase, or the synthesis phase, and that's when the DNA is replicated. We're going to spend a whole unit on that because it's so important and because it's so complicated. The next phase here, G2, that's called gap 2. It's the secondary growth phase. So that's this part. That's when you're sort of getting ready to do mitosis. Then you have mitosis, and at the very end, cytokinesis. So you're separating out the DNA here, and then you're separating out the cytoplasm here, and then look, you get 2 brand new cells. So M would be mitosis, and C would be for cytokinesis. So that's the entire cell cycle. We'll spend a little bit of time going back and giving you notes on each of these, and then notes on these, and that's the story. So interphase is not a stage of mitosis. It's the stage in between one mitotic event and the next. So it's a period of growth, maturation, and preparation that comes before mitosis and cytokinesis, or after, because it's one big cycle. So the G1 phase is the first phase. So it's the first growth phase after cell division. The cell matures, makes more cytoplasm and more organelles. The cell carries on all its normal metabolic activities. That means all the chemical reactions that it needs to live. The next phase is the S phase of interphase, the synthesis stage. So synthesis means you're making something. In this case, you're making DNA. So it's DNA synthesis. So, oops. So DNA is copied or replicated or synthesized. And so you start with one DNA molecule and you make two that are exactly the same. So these things, that's the original DNA. And the key here, whoops. And the key here is that these are identical. This is exactly the same as this. So if you start out with 46 chromosomes, you're going to replicate every single one of them and they'll be identical. The G2 phase of interphase, that's called the second growth stage, or that's the second growth phase. It's the gap2 phase. It occurs after DNA has been copied. All cell structures needed for division are made. Sometimes this is a really quick stage and not too much happens, except that you're going to make your centrioles and stuff. And both organelles and proteins are made, whatever you need. And look at, here's interphase. So the thing to notice here, so if you look at this cell, there's a little cell under it too. Sorry, I don't think I drew that right. Or you look at this cell, whichever cell you're looking at, the purple stuff is the DNA. And you can see there's not one particular clear chromosome. It's all kind of spread out. So the DNA is spread out because it needs to be read or copied or whatever. So the idea to remember with interphase is that the DNA is not condensed. It's spread out. And there it is again. The whole blue thing is interphase, which consists of G1, S, and G2. And so the key here is that before you separate out your DNA, you've got to replicate it.